Okay, so yesterday we were looking at enzymes as one type of protein, so building on the idea of biological molecules. And today we're going to look at DNA, RNA, and then a bit of DNA replication. So what we're actually going to start off with is the basic structure of DNA and RNA. So going back almost to its nucleotide form. So its nucleotide form is sort of the simplest form that we can break DNA and RNA into. So nucleotides are this sort of basic unit of DNA and RNA. And what they are, they're nitrogen containing organic substances, and they form the basis of the nucleic acids in DNA and RNA. So a nucleotide roughly looks like this. So we often draw them as a circle, a pentagon, and then a hexagon. So the circle always represents a phosphate group. So there's a phosphate attached to a nucleotide. The pentagon represents a pentose sugar. So depending on if we're talking about DNA or RNA, depends on which type of pentose sugar is present. So if we were talking about DNA, it would be deoxyribose sugar, whereas if it was RNA, it'd just be a ribose sugar. And then we have our nitrogen containing base. Sometimes these bases are actually just drawn as, rather than a hexagon, they're sometimes just drawn as a rectangle. It doesn't really matter really, but they're just these extra bits. So these are our nitrogen containing bases. And so these are the three main structures that we find that make up a nucleotide. So all these three groups together make up our nucleotide. So again, DNA, the sugar is our deoxyribose, whereas in RNA, the sugar is ribose. And all that DNA and RNA is really is long chains of nucleotides that are joined together. DNA is going to be two chains of nucleotides joined together. RNA is going to be one chain of nucleotides that's joined together. So we're going to have a look into the bonding and the structure of these a little bit more, but this is the most important bit, as our phosphate group, our pentose sugar, and nitrogen containing base. So if we were going to draw them joining together, I'm just going to talk through this bit now. So like I said, sometimes we draw the bases slightly differently. And as you can see here, the base is more like an arrow shape, like a rectangular arrow here, rather than a traditional hexagon that was drawn. You'll find them really different everywhere. It's because it doesn't really actually matter what this bit looks like. So this is our base, but our phosphate and our pentose sugar is always going to be a circle and a pentagon. Pentose because it's a pentagon sugar, pentose sugar, so it's got the 5 carbons in. So what happens is we have one nucleotide, and it has to basically, DNA and RNA are just chains of these nucleotides all joined together. So what has to happen is, to make these nucleic acids, we have to join all these different separate nucleotides together. So we make these long chains, and we actually call these long chains polynucleotides. Polynucleotides. So you can sort of see where the name's coming from, like when we talk about polymers and monomers. Monomers are single base units. Polymers are lots of those single base units joined together. Polynucleotides are lots of those single base units of nucleotides joined together. And like we said, they consist of the pentose sugar, the phosphate group, and then that nitrogen containing organic base. For them to join together, they join together over here. And it is between the pentose sugar of one nucleotide and the phosphate group of another. And so the bonds that join the sugars and the phosphates together, these are called phosphodiester bonds. And that's quite an important name to remember. The most important thing probably of this whole topic of biological molecules is knowing the names of the bonds and the correct bonds that come up. So lots of questions that will come up in exams, things like, what is the name of the bond that's formed between two nucleotides along the sugar phosphate backbone? So your answer would be phosphodiester bonds. So lots of different naming of bonds comes up, so we have to have a really good quick recall of them. These form, unsurprisingly, like nearly every single one of our bonds has formed during this topic, by a condensation reaction. So obviously condensation reaction is going to join that sugar and that phosphate together to make a phosphodiester bond. And then that will keep repeating again and again. So it's not that we just have one of them bonds the whole way down the back, we have these phosphodiester bonds. And what we basically end up doing is we end up calling this whole section that is here, just, oops. This whole section along the back, we call this the sugar phosphate backbone. So this is our sugar phosphate backbone, and that is where all of the DNA, in this instance we're going to talk about DNA for now, are held together. RNA has exactly the same bonds, phosphodiester bonds being formed in exactly the same place. Then what can happen is, if we're talking about DNA we know is made up of a double helical chain. So RNA is a single chain of nucleotides, so it's a single polynucleotide chain. And we'll talk about specifically its bases in a moment, but it's just a single polynucleotide chain. So RNA is just composed of lots of nucleotides all joined together with these phosphodiester bonds. DNA, we know, is a helical enzyme, ignore me, it's a helical protein, and it's actually a double helix. So rather than just having a single helix that's curling, we have the two different strands that curl together, and we'll look into complementary base pairing in a moment, so don't worry about that. They have a specific way of matching up, but they have to join as well. So it's not just one chain and it sits with another chain. They actually have to be able to join together. So what we end up happening is this, and I'm going to explain about the orientation of the two strands of the DNA in this moment, because as you can notice now, one strand appears to be upside down compared to the other one, and that is how this works. But our bases, so again, this polynucleotide chain joined up with those phosphodiester bonds. This polynucleotide chain again joined using those phosphodiester bonds. Let's mark them up. So we've got our phosphodiester bonds. So we've got two polynucleotide chains, and then what we need now is actually for those bases to join together. So we need something to actually keep those bases together. So between our nitrogenous bases, we have some bonds between the bases, and these are just hydrogen bonds. So we have some hydrogen bonds forming to hold the bases together. So in DNA, we have two types of bonding going on. Our orientation is a little bit different here. So what's happening, and we'll go into this in a bit more detail in a bit, but actually to make a double helix of DNA and how DNA complementary base pairs to each other, they actually run anti-parallel to each other. And this is something we'll cover in a bit more detail in a bit, but I'll pop it on now. Is DNA runs in an anti-parallel arrangement to each other. So one end is arranged in the opposite orientation to the other. And we'll talk about 5' and 3' ends in a bit. So that's all to do with numbering the carbons of the pentose sugar and which direction we run in. So we'll look in that in a little bit more detail in a little bit, but they run in an anti-parallel configuration. So what's really important is actually you don't get a chain where the pentose sugars are facing the same direction. You have one that appears to be upside down, and that's what we need to take from that now. And we'll come back to this in a bit more detail later. So we have 5 bases that we find. So we have 5 different nitrogenous bases found in DNA and RNA. And we have something called complementary base pairing. So what will happen is we have 5 bases and we can split them into 2 groups. Now, we're talking about 5 bases, actually. I want to go back a second. We're talking about them being 5 bases because we actually have some swapping, whether we're talking about DNA or RNA. So you'll have come across before A for adenine, T for thymine, C for cytosine, G for guanine, and then U for uracil. And they pair up with each other. So A and T, complementary base pair together. So an A will always pair up with a T, vice versa. C and a G will always pair up, and that is in DNA. In RNA, we don't have thymine. That is swapped out for uracil. So instead of A and T, adenine and thymine base pairing together, we have adenine and uracil. But then our cytosine and guanine stays exactly the same and they base pair together in RNA. So we split these 5 bases into 2 groups. So we have adenine and we have guanine, and these are purine bases. Now you don't need to know the actual display formula of this. I'm giving you the display formula because we'll talk about the differences. But actually for an exam, you will not be asked to draw the displayed formula for them. However, you do need to know they're purine bases. Thymine and cytosine and uracil are pyrimidine bases. So basically what you can see when you look at these is that we, when we're looking at the display structure of them, we've got adenine and guanine being made up with 2 helical structures, but thymine, cytosine and uracil just being made up of 1. And so they're pyrimidine bases. Lots of people remember them in lots of different weird ways. The easiest way, I always think, is if we go adenine, it has the number 9 at the end. Guanine has the number 9. If we count 1, 2, 3, 4, 5, 6, 7, 8, 9, you can get 9 carbons in there. They're purines. Thymine, cytosine, uracil don't have 9 in, so they are pyrimidine bases. So you have to know which ones are purines, you have to know which ones are pyrimidines, but you don't have to know how to draw the bases out. Okay, so let me just pause this for 2 seconds because this has gone to the wrong bit. Is this going to work? Probably not. Interruptions. Okay, so I just want to get something up about the nucleotides and the conformation of them. So, like we said, DNA and RNA, they are molecules that are proteins that make up everything, that's basically make up our genetic material. So DNA is a genetic material that we find in all living organisms, and they are made up of nucleotides. DNA, the main purpose of DNA is that it codes for proteins. It tells the cells what to do, and it goes along and it codes for proteins. The whole amount of DNA, the whole DNA of a single living organism, is called the genome. So when people study genomics, that's looking at the whole genome of an organism. It's looking at all the DNA of an individual organism. Now, when we're talking about eukaryotes, for example, DNA in eukaryotes is mainly found in the nucleus. We do find a little bit of DNA in the chloroplasts, we do find a little bit of DNA in mitochondria, but it's mainly found in the nucleus. Whereas if we're looking at prokaryotic organisms, the DNA is not found in an enclosed membrane. That's the main key difference of the term prokaryote and eukaryote. Eukaryote meaning true nucleus, and prokaryotic meaning non-true nucleus. It doesn't have a nucleus. It doesn't have this membrane with an envelope containing the DNA. The DNA is found in other locations. So mononucleotide is basically just our phosphate group attached to our pentose sugar, which is then attached to our nitrogenous base. So here is one there: phosphate, base, pentose. And so we have our 5 different bases: adenine, guanine, thymine, cytosine, and uracil. And remembering that uracil is only found in RNA and thymine isn't found in RNA. Adenine and guanine are large bases, so these are called purines. And the 3 other are smaller and they are called pyrimidines. In DNA, we find A, G, C, and T, and then in RNA, we find A, G, C, and U. And our pentose sugar is either ribose or deoxyribose. So that will let us know if we're talking about DNA or RNA. Now, what's quite important is when we talk about the bases and that they do this complementary base pairing, it's actually really important to realise that because A and T always pair together and C and G always pair together, we will always have the same amount of cytosine and guanine in a strand of DNA as each other because they complementary base pair together. So whatever the split and divide is between A and T and C and G, if we had 30% of the DNA being comprised of cytosine, that guarantees 30% of the DNA will be composed of guanine because they complementary base pair together. Therefore, 20% of the DNA would then be composed of adenine, and then the final 20% would be composed of thymine if we are talking about DNA in this situation. Because they're complementary base pair, we will always have the same amount of each other being present. So dinucleotides, that's when we're getting 2 nucleotides and joining them together. Like we said, we form them using condensation reactions, and that's between the phosphate group of one nucleotide and the pentose sugar of another. So what happens here is we get this bonding occurring there, and that's our condensation reaction, and that makes our phosphodiester bond. And remembering that a condensation reaction includes the release of water. So water would be a product of the condensation reaction. If we wanted to break these nucleotides apart, these dinucleotides apart to make them into mononucleotides again, then we'd go through a hydrolysis reaction, put into water into the add in water to the reaction, and then the molecules would split apart. And remembering actually that bond that we're forming is a phosphodiester bond. It's very similar to ester bonds, but we have phospho in front of it because it's because it's joining the phosphate and the sugar together, so it's a phosphodiester bond. Now, I was going to have a little talk about this phosphodiester bond stuff in a bit more detail as well. So our carbon atoms on our sugars are numbered, and they're numbered 1 to 5. So what I'm going to do is I'm going to number them 1 to 5, and they're written like this. They're written 1, and that little dash next to 1 means prime. So we'd have 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So our phosphate attaches to the hydroxyl group of our 5 carbon, and this is what we're going to talk about when we are going to make basically this double helix. And when we make this double helix, so if we we have a 5,3 phosphodiester linkage, and what this means is we have 5' end and a 3' end where they will go and join each other together, and one end of the DNA will run from 5' to 3', and the other end of the DNA will run 3' to 5'. So the phosphate attaches to the hydroxyl group of the 5' carbon of one sugar and the hydroxyl group of the 3' carbon of the next sugar, and that makes a 5', 3' phosphodiester linkage. And I can't get this to move on. It's really annoying. So polynucleotides, this is actually pretty much the same as when we were talking about our dinucleotides, but now we've just got lots of nucleotides joined together. So DNA is a polynucleotide made up from 2 long polynucleotide chains. Why is my computer not responding to anything anymore? So the 2 chains are held together in a double helix shape, and that shape, that double helix shape, that coil, that helical shape is just held together by the hydrogen bonds between the bases on each chain. So each physical chain is held together by those phosphodiester bonds on the sugar phosphate backbone, but then the bases hold the actual 2 chains together in that helical structure with those hydrogen bonds that we find between each base. So complementary base pairing. So the bases on one chain bind to the bases on the other chain. So as we've said before, adenine base pairs with thymine, guanine base pairs with cytosine. And it's actually, if we look at it, we have these shapes coming into play. There is a similarity here to that sort of concept and idea of substrate and active site. We've got shapes that fit each other, where they're going to bond together. And so what they are going to do is there's going to be hydrogen bonding between them. And so those hydrogen bonds join those bases together. So we call this complementary base pairing. Adenine will only bind to thymine. And we're saying it will only bind to thymine. There is the odd occasion where things can go wrong, and we will talk about that at another point when we look at DNA mutations. But adenine should only bind to thymine and guanine only to cytosine. So the hydrogen bonding we were talking about, it is slightly different if we're hydrogen bonding the thymine with the adenine or the cytosine with the guanine. So why each one binds with each one is actually can be seen in this diagram. So oxygen is slightly negative. So we're talking about these electrostatic charges again. So our oxygen is slightly negative, whereas hydrogen is slightly positive. Therefore, hydrogen bonds are formed between the oxygen and the hydrogen. And then adenine and thymine are only capable of forming 2 bonds, whereas guanine and cytosine form 3. So if we have a look here, we have the spare oxygen on the thymine, and that oxygen is going to hydrogen bond with the hydrogen on the adenine, hydrogen and the nitrogen on the adenine, whereas down on cytosine and guanine, we have those 2 free oxygens available. So we have those 3 bonds. So we actually have 2 hydrogen bonds formed on thymine and adenine, 3 hydrogen bonds formed on cytosine and guanine. So what we end up with is a double hydrogen bond on the top one and a triple hydrogen bond on the second one. So that is a difference. But as you can see, it's not that purines hydrogen bond together and pyrimidines hydrogen bond together. Again, it's the idea of opposites attracting. So pyrimidines and purines hydrogen bond together. So it's thymine and adenine, and then cytosine and guanine. So I want to skip past this bit actually. Don't actually want that, so let me close that. This computer is actually not doing what I wanted to do today, and show, discard them. I want to now go on to, let me open it up, looking at DNA replication for you. I'm just waiting for this to load up. My computer's being a bit of a pain. So we're going to have a look at the 5' and 3' ends of DNA in a bit more detail now. Just give me 2 seconds and we're going to look at DNA replication. Not quite sure why nothing is loading. Here we go. So we have that specific base pairing, we have polynucleotides, DNA, RNA, the anti-parallel, they run in opposite directions. And now we're just sort of going to bring in DNA replication now for the last half hour. You might have to bear with me swapping to a different slide because this looks like it's not going to work. But I'm sort of bringing in cell division now. So we've got DNA, we've got DNA inside our cells, cell division. Well, the linkage between them is the idea that actually when cells go on to divide, we know that cells cannot just split in half. Everything inside the cell has to be replicated first before the cell can be divided, before it can be split into two. So cell division, the first part of cell division, the only first thing that needs to actually happen is we actually have to have replication take place. So before mitosis can take place, we have to DNA synthesis, DNA replication. So that is what we're going to have a little look at now. And we're going to look at specifically something called semi-conservative replication. And this is actually the outcomes of sort of splitting it between A star and C. So just getting a C grade, you would need to know the method of semi-conservative replication. This isn't any different really from what you've learned. The basics at GCSE, we're just adding in some enzyme names. We'd need to know then the difference between replication and transcription. Now, this is a bit of a strange bit, point B, because transcription is part of protein synthesis, and we haven't done protein synthesis yet. So we'll actually look at that part later, but it's actually a question that does come up a lot in the exams that will say, compare and contrast DNA replication to this transcription stage of protein synthesis. And they like to use this because there is lots of similarities between the two, but they are two very different processes. So they like to make sure that you can understand the differences between them. So we will look at them once we have done protein synthesis. We'll actually spend a bit of time working through an exam question and dissecting it and what sort of answers they want. A grades, then we're looking at Michelson and Stahl, and they provided a theory of semi-conservative replication and then theory to the proof of semi-conservative replication. So, and I bet this isn't going to work. So bear with me 2 seconds. I need to enable editing. Right, so that took us back to the beginning. We just don't want it. Right, let's see if this will actually play now. Oh, enable content. Let's see if we can get it to work now. Let me get a different image up then for DNA replication. New slide. Let me just pause the recording for 2 minutes. Otherwise, you're just going to have me messing about trying to find it. Okay, so DNA replication. What has to happen then in DNA replication is we need to basically make a whole new DNA strand. And what happens is both strands will act as a template. And we call this semi-conservative replication. And we'll go into a bit more detail about that in a bit. Basically, the idea is with semi-conservative replication, one strand of the double-stranded DNA will be from the old original DNA and one strand will be new. And then for the other DNA molecule, one will be an old strand and one will be a new strand, hence the term semi-conservative. So DNA replication, we've suddenly got these words of 5' and 3' written on. And so what happens is at the end of our proteins, at the end of our polynucleotide chains, one end will actually have a phosphate group present, and one will actually have a different hydroxyl group present. So we have carbon atoms and our sugars, and then we said they've named 1' to 5'. And what happens is our phosphate attaches to the hydroxyl group of the 5' carbon of one sugar and the hydroxyl group of the 3' of the next sugar. So what would happen is, let's call this one 5'. And so what ends up happening is one end is free on either end of the chain of DNA. So we call this a 5', 3' phosphodiester linkage. But what happens is our strands run in the opposite direction. So like how I showed you before, that those nucleotide chains, when we draw them and they do the base pairings, one's actually flipped in the opposite direction, one's actually upside down. That's because the 5' end will face the 3' end of the matching strand. So I'm just going to actually, when this stops doing that, come out of this for a second and show you that in here. Get that part up again. Where has it gone? So what I mean by that is, we can actually see it quite nicely here, is our 5 carbon will face our 3 carbon of the matching strand. So let's number them. 1, 2, 3, 4, 5. So our 5 carbon will face our 3 carbon of our matching strand. So as you can see, with the way that they're numbered around differently, so our carbons will match in different directions, and that's how we get the anti-parallel. So this would be our 5' end. Oh, I don't know why it's gone way up there. But this would be our 3' end, and then this side would be 5', and then the top end would be 3'. So this is what I mean about them matching on opposite sides. And this is what we call anti-parallel. So they're running anti-parallel. If they were parallel, both top ends would be 5', both bottom ends would be 3'. But these run anti-parallel. So they run 5' to 3'. One side, 5' to 3' the other, but they're flipped around. So if I exit this one, go back to where I was, DNA replication. So the whole principle of DNA replication is to make go from one strand of DNA to 2 strands of DNA, so that then we can go through mitosis and a cell can split and we can go through cell division. So basically, we've got a DNA molecule that's in this really tightly wound helix. Now, this helix is actually really good. Like when we talked about proteins before, helical structures are great for storage. They're really tight, they're really compact. It means that they're not damaged very easily. Not great for DNA replication. So actually what we need to do is we need to unwind all the DNA. So if we have a look here, we've got an image of a chromosome and the DNA unwinding over here, and you can see that 2 new strands are being formed from the 1 strand of DNA over here. So our first stage of DNA replication is actually our DNA becoming unwound. And so I'm going to put number 1 here. This image actually shows us everything happening, and it's actually quite nice to have an image showing everything happening at the same time, because actually once DNA helicases come on and the whole process is going, it doesn't sort of stop, and it doesn't mean that all the DNA unwinds, then all the next stage happens, then all the next stage happens. They're actually happening simultaneously, and so they're sort of going constantly throughout the whole process. So it's quite nice actually to have an image where it's all happening at once. So yeah, our first stage is an enzyme called DNA helicase, and again, we know it's an enzyme because we have an ASE on the end, attaches itself to the DNA. DNA helicase is an intracellular enzyme, so it's an enzyme that we find on the inside of the cell. And what it's able to do is it comes along and it binds to the DNA. And what it does is it breaks the hydrogen bonds between the 2 polynucleotide chains. So I'm going to type this part on actually. It comes along and DNA helicase breaks the hydrogen bonds between the bases. So what happens is when these hydrogen bonds are being broken between the bases, they're the only things that are keeping this DNA wound up together in this helical shape. It's the only thing keeping it formed together. So what happens is if we break those hydrogen bonds, the helix will unwind, so it will basically split into two strands. So we get two separate strands forming at this point. So DNA helicase breaks the hydrogen bonds, which causes the DNA to unwind, and form two separate strands. So that's a really important part, is that it makes two separate strands at this point, because we can't do anything else. We can't replicate any of the DNA if we have all the strands wound together, if we don't have two separate strands. So that's our first part. So DNA helicase is actually going to basically make its way along the whole DNA molecule whilst all this rest of part of replication is taking place. So that's part one. So then what happens is DNA helicase has basically just opened up, and we've got these two separate strands. And these are our two separate strands here. We've got one called a leading strand and one called a lagging strand. We also call one a template strand and the non-template strand. At the moment, these terms such as lagging and leading aren't necessarily important at the moment. They apply more to when we're looking at things like protein synthesis, but I'll explain the difference of them slightly in a moment. So we've just got this open area of DNA, and what happens is we have some free floating nucleotides come in, and they come and join on. So for example, here we've got areas of free floating nucleotides come and join on. So for example, if we're going to call this one adenine, a thymine would come and join on here. If this one was cytosine, we would have, oh no, that one's guanine, would have a cytosine, and so on and so forth. They will all come and match up. So using the complementary base pairing principle, step 2 is free nucleotides. Free nucleotides form hydrogen bonds, again, form H bonds with complementary base pairing. Complementary base pairing. And each strand is used as a template. So each strand is having these free nucleotides coming and bonding with them and forming these hydrogen bonds. So it doesn't matter if it's the lagging strand or the leading strand. So that is what's going to happen. But at this point, we've just got two free bases sort of sat there. So I'm going to call this bit number 2. And they're not joined together, but we actually need to join them together. We need to make that sugar phosphate backbone so that we've actually got a secure polynucleotide chain. Because at this moment, we've got the hydrogen bonds holding them together to make that helical shape. We don't actually have anything keeping that nucleotide together. So what we then need to do is we actually need to reform those phosphodiester bonds to make that sugar phosphate backbone along those new free nucleotides that have just complementary base paired. So what comes along now is we have another enzyme come along. And I've just realized that I don't like this image, because this image is talking about RNA in DNA replication. So bear with me 2 seconds. This is what happens when you use something different and it opens up differently. Let me go to here. Come on, internet. Yeah, we'll use this one. I'm just going to swap the image because I'm not happy with that one. New slide. Okay, so actually this is sort of pretty much in the same place. Right. Have I got another image of DNA replication anywhere? Right, you know what? I'm just going to start drawing my own in a second because this is starting to annoy me. Try it again. Let's get rid of all them drawings. Oops. So we have DNA. That DNA is going to be unwound, and it's going to be joining on by using unwound together. We're going to draw it on here. Let's add it on here. Over here, we'll have our DNA helicase. So that is part 1. So we have DNA helicase, and that will come along. It breaks those hydrogen bonds between the bases, and it means that we have these single strands over here. Number 2, those free nucleotides that are free floating, for example, these ones here, they're all going to come along and they're going to complementary base pair with the nitrogenous bases that are already in place. And they are going to form hydrogen bonds. So I'm just going to put H for hydrogen bonds. Then what we need to do is we then need this new sugar phosphate backbone forming. And we have another enzyme come along called DNA polymerase. DNA polymerase is sort of in the name, and we're turning it into a polymer by this point. So what DNA polymerase does is this is another enzyme, and this comes along and that reforms those phosphodiester bonds that we were talking about between the nucleotides. So we would eventually get these 2 new strands. But what you can sort of start to notice is we have these 5' and 3' sort of dotting around on all these different images that I've shown. So if, for example, we show them here, our DNA polymerase actually works in different directions. So as you can see on this image, the DNA polymerase is working in one direction and the DNA polymerase on the other one is working in a different direction. Again, here, our DNA polymerase is working in a different direction to the other one. DNA polymerase works in a 5' to 3' direction. So what happens is it doesn't actually continually form. So what happens is it breaks off and we have little gaps that then have to get filled up by something called DNA ligase. And DNA ligase fills up these gaps, and we call these gaps Okazaki fragments. That's not actually essential to know at A level, but it comes in a lot, especially if you look at different textbooks that aren't officially A level textbooks, and things like Okazaki fragments start to appear, but they're not actually too much to worry about. But DNA polymerase, what DNA polymerase does is that forms a condensation reaction between our nucleotides so that we get these phosphodiester bonds forming. So it's basically just a 3-step process. It often gets overcomplicated into something more complicated than it is. But essentially, all that is really happening is, let me clear this, they're all drawings. Essentially, all that is happening is this, is we have a coil of DNA, and it starts to unwind here. And that is because of this enzyme in place, this enzyme being DNA helicase. DNA helicase comes in and it breaks those hydrogen bonds between the nitrogenous bases. Oh, there we go. This area where we are getting the two separate strands, where the strands start to separate, we call this region a replication fork. Replication fork, and that's just where it starts to split. We have those two single strands forming. So we have this, and then I'm going to draw it back like that over here and explain that in a minute. So, so yeah, let's put A, T, C, G. And remembering that they would have been complementary, so we would have a T, A, C, G on this. Oh no, G and C there. So our free nucleotides would come in and fill in the gaps. I'm going to do on free nucleotides in a different colour just to make it clear that these are free nucleotides. So our T would come in complementary base pair with our A, and they would form a hydrogen bond. A would come in, form a hydrogen bond. G, I don't think I could draw that upside down, comes in and forms a hydrogen bond, and then C. And the same on the other side, would have A, hydrogen bond, T, hydrogen bond, C, hydrogen bond, G, hydrogen bond. Then, before we can actually have them as two separate DNA strands, we can't just let go of them at this point. If we could, that would be great, but we can't. What we actually need to now do then at this point, before we can actually have two strands, is make that sugar phosphate backbone again. So I'm going to do that in a different colour. Sugar phosphate backbone needs to join between all of these nucleotides, same between these ones, and that happens because of this enzyme that's going to come along here, and that is DNA polymerase. And DNA polymerase comes along, and it does condensation reactions along that sugar phosphate backbone, and those condensation reactions are going to form those phosphodiester bonds that we already spoke about. So I've mentioned the term semi-conservative replication, and I sort of scooted past the idea that basically it's because one strand is made up of, out of the two strands in one DNA molecule, one strand's from the original DNA molecule and one strand is from the new one. So we're going to have a quick look at how it can work and what conservative replication is and what dispersive and semi-conservative is. So when DNA is replicating, we have our original DNA double helix, and if it had been conservative replication, what would have happened is we would have a completely brand new DNA strand that's made up from two DNA molecule that's made up from two brand new strands, and a DNA molecule that's made up from the two old strands. We could have dispersive, where it's literally fragmentary in between. There isn't one strand that's new, one strand that's old, it's all a mixture of both. Or semi-conservative, which is what we know to be true now, which is one strand being comprised of the old, one strand being comprised of the new to make one molecule, and the same for the opposite molecule. So that is our one that we know is true today. So what happened is there was two people called Michelson and Stahl, and they proposed this idea. They said, okay, we think that DNA replicates in a semi-conservative manner. So what they did is they used bacteria grown in a heavy isotope of nitrogen. There is two isotopes of nitrogen you can use, which is 14N and 15N. So they went and got some E. coli, and they grew this E. coli in a heavy nitrogen medium. So our E. coli DNA becomes labelled with heavy nitrogen. Now that's important because obviously all of our bases in DNA, regardless of what type of organism we are, contains nitrogen, the nitrogenous bases. So the nitrogenous bases that are going to be forming in that E. coli are going to be composed of this heavy 15N nitrogen base. So what they did is they grew this bacteria in the heavy isotope of nitrogen, generation 0, and then they took a sample of this E. coli, spun it in a centrifuge, and then they looked at where the banding of the DNA would sit, because we're talking about actually the weight of the nitrogen. So because the bases are heavy, they will sit at the bottom because of this 15N. And there was just one band of where the DNA was, and that actually sat at the bottom of the test tube. What they then did was they got the 15N labeled E. coli, and they actually got the E. coli out, and they added it to a 14N medium. So this is a light nitrogen medium. So the E. coli carries on to grow, it carries on replicating, the DNA replicates, and we grow it into a 14N. And generation 1, we take it out, we spin it in the centrifuge, and then we have a look at what then happens to the weighting. We actually find a band just in the middle, and that's because it's composed equally after one generation of heavy and light nitrogen. Wait for a second generation for it to replicate. We then have a top band and a middle band. So we have a middle band that's composed of 15 and 14N nitrogen, but we also get a top band that's made of just the 14 nitrogen. So as you can see, as the longer it's in there, we're not having any more 15N nitrogen bases being formed, because we are not in the 15N medium anymore. So that's why we have this separation. So then eventually by generation 3, we have a larger band at the top and a smaller band in the middle, and that difference would keep going up each time. So semi-conservative replication, this is basically what's happening. First replication made up of the same bands. Going to first generation, we have that replication. We have one molecule made of a new strand and an old strand, one molecule made of a new strand and an old strand. Then we get to generation 2, and actually we're having that happen, but as you can see, we've stayed with the same weight of the bases, so they're not going to change as much. So a typical answer would be something like this. Which model of DNA replication is consistent with results obtained by Mieselstein and Stahl? So what